Are macromolecules. Within cells, small organic molecules are joined together to form large molecules known as macromolecules. Macromolecules contain thousands of covalently bonded atoms, such as a protein, nucleic acid, or synthetic polymer. Macromolecules are important cellular components and perform functions necessary for the survival and growth of living organisms. The four major classes of macromolecules are carbohydrates, lipids, proteins, and nucleic acid.
Each of the four macromolecules are made up of building blocks known as monomers. Monomers are molecules that can be bonded to other identical molecules to form a polymer. A polymer is a long molecule consisting of many similar or identical building blocks linked by covalent bonds.
Creating and breaking the covalent bonds that hold monomers together is done by removing or adding water. These reactions are called dehydration synthesis and hydrolysis. In dehydration synthesis, monomers are joined by the removal of a hydroxyl group from one monomer and a hydrogen ion from a second monomer. Cells invest energy to carry out dehydration synthesis. Dehydration synthesis are anabolic, meaning they require complexity, and are endergonic because they require energy.
The covalent bonds connecting monomers in a polymer are disassembled by hydrolysis, a reaction that is effectively the reverse of dehydration. In hydrolysis, bonds are broken by the addition of water molecules. A hydrogen atom attaches to one monomer, and a hydroxyl group attaches to the adjacent monomer. Hydrolysis is catabolic, meaning it reduces complexity, and is exergonic because it releases energy.
Carbohydrates. Carbohydrates make up sugars and starches. They are made of carbon, hydrogen, and oxygen. The simplest carbohydrates are monosaccharides, or simple sugars, and are monomers of carbohydrates. The monosaccharides are connected through dehydration synthesis to form polysaccharides. Carbohydrates are used for short-term energy storage and structure.
Monosaccharides have molecular formulas that are some multiple of the unit CH2O. Monosaccharides have a carbonyl group and multiple hydroxyl groups. The most common monosaccharides are hexose sugars like glucose, galactose, and fructose. Most names for sugars end in -ose. Depending on the location of the carbonyl group, the sugar is an aldose or a ketose. Glucose, an aldose, and fructose, a ketose, are structural isomers. Monosaccharides, particularly glucose, are a major fuel for cellular work. They also function as the raw material for synthesis of other monomers such as amino acids and fatty acids. Two monosaccharides can join with a glycosidic linkage to form a disaccharide via dehydration. For example, sucrose is formed by joining glucose and fructose. Lactose is formed by joining glucose and galactose.
Polysaccharides are polymers of hundreds to thousands of monosaccharides joined by glycosidic linkages. One example of a polysaccharide is starch. Plants store a surplus of glucose as starch and withdraw it as needed for energy or carbon. Animals store glucose in a polysaccharide called glycogen. Another example is cellulose, which is a major component for the tough wall of a plant cell. Like starch, cellulose is a polymer of glucose. However, the glycosidic linkages in these two polymers are different. The difference is based on the two slightly different ring structures for glucose. These two rings form differently when the hydroxyl group is attached. If it is attached above, it is a beta-glucose, and if it's attached below, it's an alpha-glucose. Starch is a polysaccharide of an alpha-glucose, and cellulose is a polysaccharide of a beta-glucose. Another important structural polysaccharide is chitin, used in the exoskeletons of insects, spiders, and crustaceans. Chitin also provides structural support for the cell wall of many fungi.
Lipids are made from carbon, hydrogen, and oxygen and are used for long-term energy storage and insulation. Examples of lipids are fats, oils, and waxes. Fats store large amounts of energy. Although fats are not strictly polymers, they are large molecules assembled from smaller molecules by dehydration synthesis. A fat is constructed from two kinds of smaller molecules, a glycerol and fatty acids, which are lipid monomers. Glycerol is a 3-carbon alcohol with hydroxyl group attached to each carbon. A fatty acid consists of a carboxyl group attached to a long carbon skeleton, often 16 to 18 carbons long. The many nonpolar carbon-to-hydrogen bonds in the long hydrocarbon skeleton makes fats hydrophobic or repel water. Main types of lipids are triglycerides, phospholipids, and steroids.
3 fatty acids are joined to glycerol by an ester linkage or dehydration synthesis, creating a triglyceride. The 3 fatty acids in a fat can be the same or different and can vary in length. If the fatty acid has no carbon double bonds, then the molecule is a saturated fatty acid, saturated with hydrogens at every possible position. If the fatty acid has one or more carbon double bonds formed by the removal of a hydrogen atom from the carbon skeleton, then the molecule is an unsaturated fatty acid. An saturated fatty acid is a straight chain, but an unsaturated fatty acid bends whenever there is a double bond.
Phospholipids have two fatty acids attached to glycerol and a phosphate group. Additional smaller groups may be attached to the phosphate group to form a variety of phospholipids. The fatty acid tails are hydrophobic, but the phosphate group and its attachments form a hydrophilic head. Phospholipids are arranged as a bilayer at the surface of a cell. The hydrophobic heads are on the outside of the bilayer, in contact with the aqueous solution, and the hydrophobic tails point toward the interior of the bilayer. Steroids are lipids with a carbon skeleton consisting of 4 fused rings. Different steroids are created by varying functional groups attached to the rings. Cholesterol, an important steroid, is a component in an animal cell membrane. Cholesterol is also the precursor from which all other steroids are synthesized.
Proteins are the most complex biological molecules. It is made up of carbon, hydrogen, oxygen, nitrogen, and some sulfur. Proteins' function include structural support, storage, transport, cellular signaling, movement, and defense against foreign substances. Most important, protein enzymes function as a catalyst in cells, regulating metabolism by selectively accelerating chemical reactions without being consumed. All proteins are polymers of amino acid monomers joined by peptide bonds.
Amino acids are organic molecules with both carboxyl and amino groups. At the center of an amino acid is an asymmetrical carbon atom called the alpha carbon. 4 components are attached to the alpha carbon: a hydrogen atom, a carboxyl group, an amino group, and a variable R group or side chain. The physical and chemical properties of the R group determine the unique characteristics of a particular amino acid. Amino acids are joined together when a dehydration reaction removes a hydroxyl group from the carboxyl end of one amino acid and a hydrogen from the amino group of another. The resulting covalent bond is called a peptide bond. Repeating the process over and over creates a polypeptide chain. At one end is an amino acid with the free amino group, the N terminus, and at the other is an amino acid with the free carboxyl group, the C terminus. Each polypeptide has a unique linear sequence of amino acids.
The primary structure of a protein folding is a sequence of amino acids that make up a polypeptide chain. 20 different amino acids are found in proteins. The exact order of the amino acid in a specific protein is the primary sequence of that protein. Protein's secondary structure creates repeated patterns of folding of the protein backbone. The 2 most common folding patterns are the alpha helix and the beta sheet. In the alpha helix, the polypeptide backbone coils around an imaginary helix axis in a clockwise direction. In the beta sheet, the polypeptide backbone is nearly fully extended. The R groups are alternating, pointing either above and below the extended backbone. The tertiary structure refers to the overall folding of the entire polypeptide chain into a specific 3D shape. The structure of the enzyme is often a compact shape. Many proteins are formed from more than one polypeptide chain. The quaternary structure connects the different subunits that are packed together to form the overall structure of the protein. For example, a human hemoglobin molecule is made up of 4 subunits.
Protein conformation depends on the physical and chemical conditions of the protein's environment. Alterations in pH, salt concentration, temperature, or other factors can denature a protein. Denaturation caused by heat disrupts the weak interactions that stabilize conformation. Most proteins appear to undergo several intermediate stages before reaching their mature configuration.
Nucleic acid is made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The function is to store information for biological systems. There are 2 types of nucleic acids, RNA and DNA.
Nucleic acids are polymers made of nucleotide monomers. Each nucleotide consists of 3 parts: a nitrogenous base, a pentose sugar, and a phosphate group. The nitrogenous bases are rings of carbon and nitrogen that come in 2 types, purines and pyrimidines. Pyrimidines have a single 6-membered ring and are cytosine, thymine, and uracil. Purines have a 6-membered ring joined to a 5-membered ring. The 2 purines are adenine and guanine. Polynucleotides are synthesized when nucleotides are joined by phosphodiester linkages and create a repeating backbone of sugar-phosphate units.
An RNA molecule is a single polynucleotide chain. DNA molecules have 2 polynucleotide strands that form a double helix. The 2 backbones run in opposite 5' to 3' direction from each other, an arrangement referred to as antiparallel. The base pairing rules indicate that adenine always pairs with thymine and guanine with cytosine in DNA. With these base pairing rules, if we know the sequence of one strand, we can figure out the sequence of the opposite strand because both strands are complementary.
Macromolecules are important cellular components that perform a wide array of functions necessary for the survival and growth of living organisms. Thank you for watching our video about macromolecules.